I'm gonna, if you allow me, skip most of my introductory slides because by now you're kind of sick of hearing all the same intro. So, as you know, we are focusing on TDP-43 and for this talk. And regarding the background on these diseases, the point that I'm trying to make by showing you this slide is that besides all the neuropathological characterization of these diseases and the genetic components that you all know, there is this kind of very interesting continuum in clinical presentations where you have some dementia in these ALS patients that is very consistent with what we see in the FTLD patients. And also in the FTD patients, you have motor neuron disease. And some of these clinical features in FTLD are related to behavioral changes, personality languages, which in a way are very difficult to model in a mammalian system. So as you all know, this kind of continuum was nicely gelled in common molecular thread by the discovery of TDP-43 pathology. So again, this is the molecule. You all know the basics about it, but we are interested in the function of the mislocalization in the pathogenesis. So we have these NLS domains, the NES, and the functions that we already heard about. And as an important point, the vast majority of ALS has TDP-43 pathology, and around half of the FTD cases also have TDP pathology. And this is characterized by this biochemical signature of hyperphosphorylation, ubiquitination, and the appearance in, I would say, mostly in brain tissue of C-terminal fragments. And of course, you know that there is this clearance or decrease in normal nuclear TDP-43 staining in cells bearing these aggregates. So this leads to these two non-opposite pathological mechanisms of loss or gain of function. So, already very early on the discovery of TDP-43, when I was doing my postdoc in Virginia Dee's lab, was to try to see what is the role of this relocalization. So we engineer mice, transgenic mice, that overexpress TDP-43 form that has a mutation in the NLS, so it will go into the cytoplasm. And this was described in a 2008 paper. So our system is a biogenic system, so we have the advantage of having temporal control by using doxycycline. So you have a TET-off system where when you have the drug present, you suppress the expression of the gene. And you also have cellular specificity by using a driver line with CAM kinase 2 alpha promoter, which drives the expression to neurons, and more specifically enriched in forebrain neurons, but not exclusively. But the motor neurons, the lower motor neurons, are not expressing this protein. So the timeline is very important for us because we wanted to make sure that we don't have any developmental defects or early postnatal defects. So what we decided to do very early on was to repress the expression until weaning. So we did the weaning at P28, and then we studied at different time points after that release of suppression. So just a summary of what we found in the first characterization of these mice is that we have a very rapid, for what transgenic animal models of neurodegenerative diseases time courses, is a very rapid neurodegeneration in selective areas. Here we have HNA staining of hippocampus, and we can see that the dentate gyrus is really, really rapidly degenerating, but CA1, CA3 regions are relatively spared. Here we have a staining of TDP-43 in a non-transgenic, in the nuclear fraction, and here we have the nice cytoplasmic staining. And by Western blot, we checked that the areas that were expressing the protein were what you would predict from the CAM kinase promoter. So basically, you have a lot of expression in cortex and hippocampus, and some in the olfactory bulb, but nothing in the spinal cord, and a little bit in the rest of the brain. And one interesting thing that we saw was that there was corticospinal tract degeneration, which is something that is very typical of TDP-43 proteinopathies, and this is revealed by phosphoneurofilament staining here. And also we have gliosis, astrogliosis, revealed by GFAP staining, but we have no evidence of muscle atrophy. And we wanted to see a little bit more about the gliosis phenotype, and there is a region-specific, so it's related to the areas where the transgene is expressed. As you can see here, then the gyrus and the CA1 regions have normal staining here, and profound astrogliosis in these panels. The same in the cortical area, but the cerebellum, which does not express the transgene, is spared. And here is the double immunofluorescence for human TDP-43, because I wanted to remind you that this is a construct that is untagged, because we found that even short tags like MIC can change the solubility and degradation of TDP-43. And you can see here that by GFAP, you have, again, a lot of cortical astrogliosis, and you can see here by the overlap that they actually neurons that are expressing, not astrocytes. And something similar you can see with the microglia. By IBA1 staining, you see the staining here that is very much increased in the transgenic mice, and you have a proliferation and differentiation of microglia. And lastly, we also analyze by microarray analysis samples from cortices of these mice at early time points, around 2 weeks after expression, so we can see kind of primary changes in gene expression. And we found that these blue squares are the NLS mice, and they group together very far away from the other control and wild-type mice, the TDP wild-type mice. And they actually have a very different pattern of gene expression dysregulation. So that was published work. I want to tell you about some of the unpublished work we've been doing in my lab now. So the idea is, so we say that this TDP-43 mislocalization is a hallmark of the disease, and we shown that in these mice we have all these changes that are evoking neuropathological features of TDP-43 proteinopathies. So we wanted to have a much more detailed behavioral phenotyping of these mice, so we can see if we can use it as a relevant model of mostly FTD because of the pattern of gene expression. And this work that I'm showing is mostly done by a very talented PhD student, which I have a completely undisclosed photograph that I stole from him here. So just general things, the animals look pretty normal in the cage. You don't see a lot of phenotype when you see them in the cage running around, and actually you can see here a weight curve of the animal, and you can see that in the transgenic mice up to around 1 or 2 weeks after induction, they have the same weight. They have a small dip afterwards, but then the growth curve is pretty much slope, so the animals are really a little bit smaller, but not very different, and they grow normally. But what we wanted to see if there was a gross atrophy of the areas that we kind of hinted that there was some degeneration going on in the previous study. So we measured, just very crudely, the cortical and the hippocampal weight in these animals, and we chose 2 time points for this analysis, 1 month and 6 months after expression. So you can see that in both areas, by 1 month you don't have differences in the weight of this structure, but by 6 months you have significant degeneration. And another thing that we tested as a general thing, because it's important for the behavioral test we're going to address later, is olfactory function, because we know that this protein expresses in the olfactory bulb. So we did this habituation test, which involves both social and artificial smells, and we can see that both transgenic and control animals are indistinguishable. Okay, so I wanted to combine two things that we did at the same time, so for the matter of time. But we wanted to ask first, what are the phenotypes in different domains in these animals at early time points, around 1 month. And also, we wanted to address what is the effect of suppressing the expression of this transgene, because nobody knows if you stop the overexpression of TDP-43, if you can change the course of the pathology or the in this case, the behavioral changes. So we devised this protocol where we have two groups, which of course have controls and transgenic mice within each one. We have one group that we call 1 month, which is the 1 month induction that I showed you before, after weaning. And the other group was 2 weeks of induction, and then we have 2 weeks of suppression. So we only have 2 weeks of transgene expression, and then we suppress it, and we compare it at the same time at 2 months of age, which is 1 month post-weaning. So we wanted to see if this system was working in our hands, so we did both double immunofluorescence for total and the transgene TDP-43, and see that the transgene is turned on and off nicely at these time points. We can see the cytoplasmic expression here and the normal nuclear staining. We did checking by Western blot too, and it represses to normal levels by 2 weeks. So another thing that we were interested in was not only if you have a phenotype early on, but also if you can distinguish between prevention or reversion of the phenotype, because it's not the same if the animal already has a phenotype and you can revert it, that if you just stop the changes so it doesn't get it. So we had to analyze the behavior at 0.5 months. So for that, we start with the motor domain of behavior, and we use both fixed speed and accelerated rotor, which is a measure of motor coordination and balance. And we can see here that in the transgenic animals, you have a profound effect of in this test for motor function as early as 2 weeks after induction. And surprisingly, we tested the animals at 1 month. Of course, the 1 month group, which is down here, is completely also bad at doing this task. But we can reverse the phenotype when we suppress the transgene by 2 weeks. Another measure of motor impairment we use is the clasping analysis, which is a measure of spasticity in mice. And this is a pretty robust phenotype in these mice, because this is the time course of the phenotype. By 2 weeks, you have like 80% of the mice doing it, and then all of them. Actually, we call them like a cheap PCR, because you lift them by the tail, and they do this clasping phenotype, and you know that they are biogenic animals. So the phenotype in these animals is that by 1 month, as I told you, all of them make this clasping, but it also is repressed. I'm sorry, it also is repressed significantly by two weeks of suppression. Another thing we wanted to address is the cognitive domain, and we did several tasks. I'm showing you one now, but we did a Y-maze test, which is a test that addresses working memory and spatial memory too, because it is based on the natural tendency of the animal to not go into a familiar place. It tries to visit different arms of this maze. So the normal animals, you can see here, will have a different than 50% alternation. Means that it remembers that it went to a specific arm and it will go to a different one. So if 50% is just chance level, and that's exactly what happens in the transgenic mice. It doesn't remember that. So that happens at 0.5 months. Of course, it happens at 1 month too, but again, we can reverse this phenotype by suppressing the transgene by 2 weeks of dog's treatment. And importantly, this is controlled by the total entries in the arms, because you have to check for mobility, anxiety, motivation, and that, and this is non-significant differences. And the third domain that we were really interested in, because actually there is no report of that in TDP-43 mice, as far as we know, is social behavior, which is very affected in FTD. So in this task, what basically is, we're trying to analyze how how much the animal likes to interact with a partner, right? And by 0.5 months, the phenotype is already present. You have social deficits in these mice, which are more profound by 1 month. And surprisingly, because the other two domains were reversible, we couldn't reverse this phenotype of social defects. And again, we have some internal controls with the exploration time in the social chamber, meaning that you don't have problems with anxiety or perceiving the stimulus as aversive or something. So we wanted to see how this correlates with neurodegeneration, because as I said, there was degeneration, but at early time points, it was non-detectable by these gross anatomy measures. So we did a little bit more subtle measurement of cortical thickness by Nguyen staining, which is a neuronal marker. So quickly, we kind of confirmed that the dentate jar was very susceptible to TDP-43 expression. But we measure also somatosensory cortex and motor cortex, and we found no changes in this parameter. So again, cortical thickness is also a little bit crude for level of analysis. So what we did is neuronal counts of Nguyen positive cells, and I'm just showing you here some examples of the somatosensory cortex, but below is the quantification. And we use somatosensory cortex, motor cortex, and prefrontal cortex, which are all involved in these tasks. And we find that by 0.5 months, there is no signs of neurodegeneration, but we have a profound phenotype. By 1 month, we have, in this case, a little bit of a mild decrease, which is not significant, but it's clearly a tendency in the motor cortex and the prefrontal cortex. The last part is we want to see if this happens when the animal is really degenerated. We know about 6 months that you have like a 20 or 30% loss of tissue, of cortical and hippocampal tissue. So we wanted to see if you can reverse these phenotypes when the animal is older and is more degenerated. So we checked that all the parts are working with the same techniques by immunofluorescence. And you can see how the hippocampus is very much degenerated here, but the system is expressing and then is repressible to normal levels. And we analyzed this domain that was really changing the motor domain, and we can see that in this case, when the animals are really degenerated, we cannot reverse this phenotype, either the rotorol or the limb clasping, which is 100% penetrant. So to finalize this, we did the same measurement of cortical or granule cell layer thickness, and we can confirm that here we have profound degeneration. We have like a 20 or 25% loss of cortical volume, and in the case of the dentate jars was like an 80% decrease. It's very degenerated. So to sum up, we wanted to perform a battery of behavioral tests to better characterize these mice, and we found that when you suppress, when you induce the transgene by removal dogs, you have motor abnormalities, and we have several tests that I didn't have time to show you in each domain. You have cognitive problems, and you have remarkably social deficits. And to see if these were reversible, we did this 2-week suppression of expression by doxycycline treatment, and we found that some of these aspects were reversible, the cognitive and motor aspects, but not the social. So when we treated these older mice, we could not reverse these phenotypes. So what we are proposing is that, one, I think we have an animal model that could be interesting to analyze some of the more FTD-like behaviors that we cannot address when you have a pan-neuronal constitutive model, because it has this lower motor neuron impairment and paralysis and eventually death that you don't have here. And second, I think this reversibility and this really early on phenotype tells us that the important thing is not the degeneration in this early time point, but is the functional changes in the neurons that are still in the circuit. When you have too much damage to the circuit, like in the later time points, you cannot rescue them. So I just want to finish by thanking the place where I did my postdoc and we started this animal work, and the people in my lab, and some collaborators and the funding agency. Thank you.